On this week's podcast, I'll be presenting on two studies that each contribute to the evidence of gene-environment interactions in autism with influences from both the father and the mother. They shouldn't be interpreted as a blame one of the parents scenario, but rather with the lens of that we know more about windows of susceptibility for environmental risk factors than we did before, and potential but not documented prevention measures. Don't believe the headlines that you've been reading that either of these studies actually demonstrates a reduction of risk. So far, there have been a few studies looking at the risk of autism born to mothers with diabetes. In fact, last year there was a meta-analysis which combined the data and compared and contrasted them. So this concept isn't new. The meta-analysis showed a consistent but a mild increase in risk following diabetes in the mother, but the approach had a weakness. In defining diabetes, they included type 1, type 2, and gestational diabetes altogether. Current thinking is that type 1 diabetes is rooted in genetics. Type 2 is caused by both genetic and environmental factors. Gestational diabetes is more of a mystery, but probably a combination of both, including obviously hormone levels. Gestational diabetes starts during pregnancy and most of the time resolves after the baby is born. A new study out of California was large enough that it had sample sizes of both women who had type 2 diabetes and gestational diabetes separately, and they were also able to control for confounders like maternal age, smoking, obesity or weight gain, preeclampsia, or other issues during pregnancy and premature birth. Because this information sits in a huge database of patients who are covered by the Kaiser HMO in California, precise data was able to be collected on the diagnosis of autism, diagnosis of diabetes, the timing of diagnosis, and medication use. This kind of resource has been enormously valuable to study medical conditions and risk of autism, as well as comorbidities associated with autism. So if you were to look at the raw data, it looks like both types of diabetes are associated with increased risk of autism. However, when you take into account the confounders, especially maternal age, which is independently linked to autism, only gestational diabetes led to an increased risk. It was about 45%. Also, if the gestational diabetes was diagnosed after 28 weeks of pregnancy, there was no more risk. So it only occurred if the diagnosis of gestational diabetes was made during a certain window of gestation. These analyses may explain inconsistent findings on risk of autism in mothers with diabetes that were shown in previous studies. Because gestational diabetes is associated with an increased risk, but type 2 diabetes is not, the authors speculated that some children in the group with gestational diabetes diagnosed at 26 weeks or earlier may have been exposed to untreated hyperglycemia during critical brain developmental windows, which led to ASD risk after birth. Pre-existing, that means known type 2 diabetes, may have been able to be treated aggressively during pregnancy, which may have reduced the effect on fetal brain development. But, that's just an idea. To be clear, there's no definitive evidence here to say that catching it any earlier or treating it any differently would have changed outcome. And the risk was statistically elevated, but not in the range to say that it's a definitive cause of autism. This study could not look directly at a gene-environment interaction, but they did do something creative. They looked at the risk of children with an older sibling with autism. There were actually a few with an older sibling whose mother also had diabetes. This didn't further increase the risk of autism, but at least they looked. The study provides even more evidence to obstetricians that gestational diabetes needs to be managed carefully. The diabetes medications didn't affect risk, so there is something about the gestational diabetes, not the medications used to treat it, that is leading to this increase in risk. Some cases of gestational diabetes can be managed with diet alone, so it's important to talk to your OB or other healthcare professional and be tested if you're pregnant. Okay, so I promised you a study about father's contribution of risk. I've also been harping about the concept of epigenetics in autism. So a study out of Johns Hopkins School of Public Health looked at sperm of fathers of children with autism to catch any differences in methylation in the DNA of sperm. It was part of an NIH and Autism Speaks funded research study that looked at the pregnancies of mothers who already had one child affected with autism, so their risk of having another child was higher. Why methylation? Adding these methyl groups to DNA does not change the sequence, but it can turn on or off the transcription of genes. The epigenetic code has been called the ghost in the DNA and the actual second genetic code in addition to what's already being captured by traditional methodologies. It's also a highly researched area recently because of the potential role of gene-environment interactions in autism. Certain environmental factors like diet, some chemicals, and other things have been known to affect methylation on the epigenome, which can last for several generations. The goal of this study was to examine areas of the DNA in sperm that are differentially methylated. That is, they have more areas of methylation on the DNA. And more importantly, where those methylation spots are happening. On what genes? Are they related to autism? What is really interesting is that they compared the DNA methylation points in sperm to those seen in human brain tissue. How are they able to do this? By having brain tissue from people with autism to study. The findings of this study are pretty amazing on their own, but strengthened through the comparison of findings on human brain tissue. So what they found is a strong relationship between DNA methylation in sperm from fathers and the 12-month ASD-related phenotype. The study doesn't have an actual diagnostic information yet, but thanks from funding from Autism Speaks, they will. The degree of methylation change was quite substantial compared to recent reports that looked at the epigenome. And the investigators looked at the genes where DNA was methylated, and they mostly had to do with brain development and neurogenesis. They were also very close to patterns in the human cerebellum of individuals with autism. So this provides independent corroboration of the importance of epigenetics in autism, and while not examined in this analysis, influence of environmental factors which may be converging on DNA expression to produce autism symptoms. Of interest, the methylation patterns were not similar compared to the cortex of individuals with autism, but that doesn't influence how exciting the findings were. In the third week of Autism Awareness Month, what are some tips on how to support autism research? Well, in light of this new science on how researchers are relying on human postmortem brain tissue to understand autism, one thing we can all do is register to donate our brains after we die to the Autism BrainNet. It's easy, it's non-binding, and you'll get a very informative newsletter every few months updating you on the progress of research in this area. Go to takebrains.org to register. Talk to you next week when I'll have a special report from the ASF Day of Learning. Talk to you then.